Welcome to the Translational Clinical Pharmacology webinar in cell and gene therapy. My name is Devin Welty, Vice President, Clinical Pharmacology at Allucent. In this webinar, I'll discuss the tremendous growth in this field and the challenges that are accompanying that growth. And then secondly, how clinical pharmacology is working in the area of translational aspects for AAV and CAR T cell therapies to improve first-in-human dose. The tremendous growth in cell and gene therapy is fueled by these transformative, curative, and durable effects for unmet medical needs that we see almost on a daily basis in the news for children who are hearing for the first time in their lives, or patients with sickle cell disease that no longer experience painful vaso-occlusive crises, or a patient that is looking at a future without blindness. Currently, there are over 800 INDs under review at FDA, 2,000 clinical trials ongoing with 200 in phase 3, and in 2023, 7 cell and gene therapy products were approved in the US, and the FDA is predicting 10 to 20 approvals per year by 2025. Along with this growth have come challenges, and this was revealed in a March 2023 congressional inquiry letter to Dr. Peter Marks, the director of CBER, requesting a response to the increased number of INDs and clinical holds for cell and gene therapy products, which was brought to light by an investigative article in the Wall Street Journal that found between 2014 and 2021, there was a 43% increase in INDs and a 66% increase in clinical holds. And of these clinical holds, 90% were generated in CBER, 40% on cell and gene therapy studies. Looking closer at the reason for the clinical holds, they were primarily driven by clinical safety, whether it was for CAR T-based therapeutics or lentiviral or AAV or other cell and gene therapy products. And this was followed by CMC and preclinical findings. The most common reason for a clinical hold was an adverse event or patient death. And on average, these holds delayed the development program by 6 months, although up to 19 months in some cases. The purpose of this discussion is to understand how we can potentially mitigate these clinical holds based on considerations for starting dose. And there are two ways to think of the starting dose, and one of them is an ethical aspect, and that is first-in-human studies are primarily in patients, often in pediatric populations with unmet medical needs, and there is the expectation for clinical benefit in addition to safety. It's a fine balance. And secondly, there are invasive routes of administration that often require surgical procedures. For instance, administering into the brain requires neurosurgery that increases the risk to patients to be considered in the benefit to risk equation. And third, there are high potential for immunogenicity that prevents readministration, and it is therefore a one-shot on goal in most cases. There are exceptions to that. But Juvic, which was approved last year for dystrophic epidermolysis bullosa, is administered on a weekly basis by the patient to their skin. And this is due to the use of a herpes simplex virus vector that evades the immune system. And then finally, as a single administration, there are prolonged biological effects that the patient lives with for months to years. There's also a business reason for getting the starting dose right, and that is encapsulated in this comment from Emily Walsh, a principal at Tremont Therapeutics Consulting, in which she notes that if the first cohort shows signs of efficacy in a first-in-human trial, it generates interest from the patient population, from caregivers and investigators, and the trial may therefore enroll faster and reduce the overall costs of the trial. However, if the dose is too high and there are adverse effects, that can cause a clinical hold and or having the product go back into preclinical development to better understand how to dose it in humans. So looking at the ways in which clinical pharmacology are now involved in translational aspects, we'll turn first to AAV gene therapy. In the guidance early phase clinical trials for cell and gene therapy products, in the section on starting dose, it's noted that allometric scaling, which is useful for small molecules and biologics, may not be valuable for cell and gene therapy products, and that traditional PK and pharmacodynamic correlations might not be possible. Looking at the complexity of an AAV administration, there is commonality between other modalities, and that is the dose can be measured in vector genomes in the capsids. The exposure for a dose exposure response relationship is described as the tissue transduction. That is, the vector genomes that have been inserted into the nucleus, either in an episome or integrated into the genome, as vector genomes per microgram of genomic DNA. This exposure can be related to the expressed protein and the response to develop an exposure-response relationship. And it's important to note that the expressed protein itself may be able to be considered as a surrogate marker for the ultimate clinical endpoint. And this was the case with the recent approval of Elevidys, Sarepta's product for Duchenne muscular dystrophy, in which the review of the data revealed that there was an association between the microdystrophin that was expressed to the clinical response, and therefore this product went under accelerated approval for that reason. In order to really understand the pharmacokinetics and pharmacodynamics, though, it's important to look at the underlying biology of tropism, where AAVs are attached to receptors on the cell surface to be endocytosed to traffic to the nucleus, and the whole transcription, translation, and protein export aspects of gene therapy. And these factors can be both cell and species specific, which can be integrated into something like a systems pharmacology model, which is able then to scale these processes in addition to dose exposure, exposure response relationships for scaling to humans. Then if we look at the different approaches for dose scaling and starting dose in first-in-human gene therapy trials, the first is allometric scaling, and as noted in the guidance, uses a dose response information in preclinical studies. It scales the dose for systemic administration, typically by body surface area, and local administration by tissue volume. However, it assumes tropism, transduction efficiency, protein expression, and kinetics are similar across all species. Next up is the dose exposure response relationship, or PK/PD. And this empirically relates the dose to exposure to protein expression and response. It can detect species differences in the transduction efficiency and protein expression to predict human. However, it lacks the mechanistic understanding for species or patient population differences. And then finally, mechanistic modeling employs a systems pharmacology approach to scale all of the biological systems and processes like tropism and translation to make assessments of dose across species. In addition to this, physiologically based pharmacokinetics is also important for scaling distribution of cell or vector to tissues, which may involve differences across species. Together, this can be used to create virtual patient populations to look at the properties of the asset as related to how it would support the disease population. All of this, though, involves changes in the complexity and cost, and oftentimes it's a factor of wanting to get into the clinic, accelerating that into the clinic, and sometimes these can be overlooked in preparing for that first in human study. And this was the case in a recent project that we worked on for an AAV therapeutic administered into the brain for treatment of a neurodegenerative disease. In this program, a mouse disease model was used to define the minimum effective dose. Non-human primate tox studies were used to identify the no observable adverse effect level dose. These doses were then scaled to a human equivalent dose based on brain volume. And the advantage of this method is that it was very easy to scale across species. And the disadvantage is that it assumes all of these processes, such as diffusion in the brain, uptake into the brain tissue, translocation to the nucleus, transduction, and expression of the protein, were all the same from one species to the next, which is very unlikely in a biological system. The outcome of this brain size allometric scaling was that the product lacked a safety margin to proceed into the clinic. And that is, the mouse minimum effective dose scaled to a human brain was higher than the monkey NOAEL dose scaled to the human equivalent dose, and this created a safety margin below 1. At this point, the sponsor approached Allucent and asked if there were other ways that they could consider in scaling for a first in human dose. And when we began to look at the data, we realized that the sponsor had very rich non-human primate tox studies, and that it was possible for us to create an exposure response relationship, that is, the vector genome copies per microgram of DNA in different regions of the brain at different time points and across 2 different studies, and compare that to the percent protein knockdown from baseline. And we were able to establish, using a PK/PD approach, that a concentration was achievable to meet the 20% protein knockdown that was considered necessary for minimally efficacious response. We could then look for a dose exposure relationship of a dose that would give us this concentration. Using the same data as before, we noted there was a linear relationship between the dose and the exposure in different regions of the brain. And by establishing this, we could then identify a dose that would achieve the 20% knockdown exposure that was needed. When we put this PKPD model to work, we found that we could establish a first-in-human dose that would provide a minimally efficacious protein knockdown with a safety margin of over 100. Further, with this approach, we could establish a dose escalation, not just based on dose alone, but also based on potential protein knockdown, and give us a hint of where we were headed with respect to a therapeutic effect and where we would achieve a balance between maximal protein knockdown and beginning to reach a safety margin of 1. When we presented this to the FDA, they agreed with the approach to enable the study to start. And this is one of the advantages of a sparse guidance, is that it creates the opportunity for greater scientific discussion and justification and rationale for a starting dose. And this was agreed upon then with both the sponsor and the FDA. Secondly, we found the learning from this was that the transduction efficiency was much greater in a non-human primate brain than it was in a mouse. Now I'll turn to translational aspects clinical pharmacology is adopting for the CAR T cell therapy field. In the recently issued guidance on chimeric antigen receptor T cells this year, a similarly sparse starting dose segment is provided, in which it comments that previous clinical experience is valuable. However, that the in vivo behavior of CAR T cells may be different depending on the disease, antigen load, study population, and CAR constructs. So that's a lot to consider. When we look at the 6 approved products for hematological cancers, we note that the innovator, Kymriah, first in class, the dose, clinical starting dose, and those of the other products that have been successfully developed are all about the same. Notably, the preclinical doses that show efficacy in a mouse tumor model are far higher than the starting clinical dose. And therefore, there's a couple of reasons why this could be. And first is that it's an objective in the preclinical studies to give high doses to show efficacy and move the program along. Secondly, the immune system in mice is much less sensitive to that in humans. Therefore, this preclinical information is not so much supporting the clinical dose as it is to demonstrate activity. The clinical starting dose is based more on clinical precedence. And why is it so challenging for us to establish scaling from preclinical studies to the clinic? And the reason is that CAR Ts are considered a living drug, and that is these T cells, which are modified with the transgene for a chimeric antigen receptor to a target on tumor cells and also normal B cells. Once they attach, these co-stimulatory domains begin to initiate T cell proliferation, cytokine secretion, which is what kills the tumor cell, and other cytolysis mechanisms. Therefore, CAR T cells expand, they migrate, and they persist in patients to varying degrees, depending on the in vivo environment and on the immune functions. So CAR T cells have a different way of expressing kinetics, but it's very similar to small molecules and other biologics. In this paper by Mueller, it's expressed in two ways. First, by expansion, and that is soon after administration of the T cells, there's a drop, which is considered a redistribution phase, followed by a rapid expansion that meets a Cmax based on PCR at around 2 weeks post-infusion. Secondly, there is an AUC 0 to day 28 that's calculated, and this is due to the fact that the first response measurement is usually at day 28 following administration in the patient. This expansion phase is due to the CAR T cells meeting up with the B cells, both the diseased and normal B cells, and growing. These CAR T cells are growing as a response to meeting up with the B cell targets. Once those B cell targets begin to diminish in the patient due to the cytolysis, the CAR T cells begin to decline as their substrate is beginning to decline. Then they start to change and transform from an effector T cell to a memory T cell. And these memory T cells then can persist out to up to 2 years and even beyond. So this is the persistence phase of CAR T cell therapy. Then if we look at how disease burden affects the kinetics, safety, and efficacy in a patient population in which the disease burden is stratified from low disease burden to greater than 50% disease burden, bone marrow percent blasts, we find that the Cmax is driven by this disease burden, so that these points in gray are the Cmax values. At lower disease burdens, the Cmax is lower. At higher disease burdens, it increases. So same dose, different Cmax values, all based on this disease burden, which feeds and proliferates the CAR T cells. Secondly, these values and numerical values within each one of these stratifications indicates the incidence of the most severe adverse effect in CAR T cells, cytokine release syndrome. 0 is mild, 4 is life-threatening. In the low disease burden patients, the CRS is manageable. Once the disease burden gets higher, the incidence and the severity become more problematic in these patients. Therefore, it's again the response of the CAR T cells releasing cytokines in response to the population of B cells that it's attacking. More B cells, greater cytokine, greater potential for cytokine release syndrome. And finally, we can look at clinical response and how it is correlated to disease burden and not to exposure. So in all of these stratified patients, from low disease burden to high disease burden, the clinical response is very good, both clinical response and clinical response with incomplete hematological recovery. The B cells have adapted what they need to achieve to fight these disease cells. And therefore, it's not associated with Cmax, but to the disease burden itself. Knowing all of this, then we can begin to build models, systems pharmacology models that incorporate both the intrinsic properties of the CAR T cells that are dosed to the B cell disease burden population, and how the response of the CAR T cells in releasing cytokines to fight the B cells occurs. And as the B cells decline, how the CAR T cells begin to change to a memory phenotype. This model was built with just a few patients, but then it was verified in a larger patient population where the investigators simulated the Cmax and the AUC and compared it to the observed Cmax and AUC and found a good correlation between their predicted and their observed outcomes. Therefore, this is a way then that CAR T cells can be modeled to give us a better understanding of how they behave in a disease state. And this is very important, although obviously the approved products have all used clinical experience to set their doses. We're now beginning to move beyond just hematological cancers and now moving into solid tumors. And what's becoming evident with solid tumors and CAR T cells is that the dose may need to be actually higher than a mouse tumor model dose because the distribution of the T cells to the tumor microenvironment are different in humans than they are in the mouse tumor model. Physiologically based pharmacokinetic modeling can help describe what kinds of doses and how much higher they need to be than the mouse as a way of supporting that starting dose. Secondly, CAR T cells are expanding rapidly beyond oncology into autoimmune, inflammatory diseases, CNS, metabolic endocrinology, and infectious diseases. Therefore, it's hard to use a clinical precedence when new diseases are being approached with these CAR T cells. Having that systems pharmacology approach used as the basis for establishing doses based on disease burdens in these other patient populations can be invaluable. And finally, even within hematological cancers, there are changes that are occurring, for instance, in the short manufacturing processes to get the treatment to the patient faster. And sometimes that's a critical point in the patient survival. And therefore, these faster manufacturing processes change the CAR T cell properties slightly. And these can be incorporated into a systems pharmacology model as to how they would be and how they would respond to a cancer environment. In summary, cell and gene therapy is rapidly advancing, and it is expected to dominate future therapeutic markets. The importance of translation and starting dose for cell and gene therapy products is critical for ethical and efficient and successful development programs. There is limited regulatory guidance for translational aspects of cell and gene therapy products to a starting dose. And this is where innovation and scientific justification and rationale plays a big part in partnering with the FDA and other regulatory agencies and the sponsor. And then finally, clinical pharmacology is adapting these innovative model-informed approaches to better translate preclinical data into starting doses for these cell and gene therapy products. So with that, I would thank you first for attending this webinar. Secondly, if you have questions, please reach out. We are more than happy to discuss any potential projects that you may have where we can support bringing a successful product for you into the clinic. Thank you very much.